Hey, I'm Charlie Hind. I'm a senior scientist and project team leader at UKHSA, and I work in the antimicrobial discovery, development and diagnostics team. So I manage the Open Innovation Platform at UKHSA, and we've developed this over the last 9 years or so to work with multidisciplinary collaborators, mainly from academia and small and medium enterprises, to enable drug discovery against infectious diseases. And we have a specific interest in the gram-negative and gram-positive bacteria, priority pathogens, fungi, and high-consequence viruses. And as part of the Open Innovation Platform, we host panels from multiple collaborators and from within our own team as well. And researchers can access these panels of bacterial and fungal pathogens to test their own drugs against them. As part of the Open Innovation Platform, we're now happy to be able to host the mini panels from the CF AMR Syndicate. And these are panels of Pseudomonas aeruginosa, Burkholderia complex, and non-tuberculosis mycobacterium, which are all well-known CF causative agents. The panels are genetically diverse and they are clinical isolates from CF patients. The main challenges that this mini panel is designed to address include using CF-relevant isolates from the early stages of drug discovery. So we often see examples where drug discovery has happened on reference strains or lab-adapted strains, and these are not relevant to the clinic, to the final disease stage that you're trying to target. So we think it's very important to test early on in clinically relevant isolates and in clinically relevant media. So for cystic fibrosis, this would include the mini panels, and it would include testing in cystic fibrosis mimicking media. So the cystic fibrosis mimicking media is much more complicated than traditional microbiology broths. So it's to mimic the environment in the lung, and you can have versions that are sputum-like and non-sputum-like. And testing in these is really, really informative to the drug development pathways. So you'll see a lot of examples of FDA-approved drugs that work really, really well in traditional broths, but are useless in cystic fibrosis mimicking medias. They're not used for cystic fibrosis, and you can see why when you do this sort of testing. Researchers can get in contact with us with our email address. That's to initiate discussions, and then there are either two pathways that we generally go down. So either the researchers can send their compounds to us and my team will test them against the panels that we host, or especially if there is a multidisciplinary training aspect to this, they can actually send a person, a student or themselves, to bring the compounds to the lab themselves, get training in the relevant microbiology techniques so they can test their own compounds against the panels. The Open Innovation Platform was actually given a remit by the Department of Health and Social Care back in 2019 to focus on non-traditional therapies. So since then we've really developed our capabilities in this area, incorporating flexible techniques and trying to build the relevant techniques, because the traditional MIC is not always the right way to measure efficacy for non-traditional. Since then, we've worked a lot on bacteriophage, lysins, antimicrobial peptides, microbiome modulators, antibiotic resistance breakers such as adjuvant therapies or potentiators. And we test these alongside more traditional approaches such as rationally designed small molecules or repurposing drugs from other areas.